All right, so everything that you need for the cheek cell lab is in the beakers that I gave you. Again, just like the strawberry lab, you have this hook. I already cut them off for you, so you don't need to do anything with that. The test tube, the empty test tube where you will be pouring everything into. Your lysis solution, but we're not going to be using the 10 milliliters that are in here. We're only going to be using 5. So that, in other words, we're only pouring half the contents out, okay? Everybody understand that? Yes? All right. And then you have a little thing. This will be the last thing you use. Here, what you're going to do is when your DNA, you see your DNA and it's precipitated out, you're going to come up to me with this. I'm going to fill it with alcohol, and then you're going to go back, scoop out your DNA with this, and then you're going to put your DNA in here, in the alcohol. You might have to move the thing around a little bit to knock it off. Seal the cap. Okay? Couple things I wanted to point out, just a reminder. Remember with the alcohol, the appropriate way to pour the alcohol is to, at an angle, very slowly, because remember, you don't want to mix the layers. Remember, the purpose of this lysis is to break down the cell membrane, allowing the contents, including the DNA, to spill out into the solution. Any questions? All right, so your first step is what you need to do now, before you rinse your mouth out with the water I provided you in the cup. You want to scrape with your teeth, kind of a little bit of scraping on the inside of your cheeks. Okay, you want to shed off some of those cells. All right, once you've done that, I'll have everybody pick that cup up at the same time, put it in their mouth at the same time. 30 seconds swishing around, just like it was mouthwash, really flushing it around. After 30 seconds, I will tell you to spit the contents out into the cup. All right, you're going to go ahead, doing your best not to spill it. You're going to pour it into this test tube here. You're going to pour the lysis. How many mils? 5. So there's 10 milliliters in here. You're only pouring 5. So my suggestion, pour slowly so you don't overpour. Once you've done that, you're going to cap your thumb at the top of the test tube. You're not going to shake. You're going to do this for a period of about 2 minutes. You see how very slowly I'm doing it, right? All right. And then at that point, the next step would be to pour the alcohol in to precipitate out the DNA. However, this time, after we pour the alcohol in, you're going to put it back in the beaker, and we're going to have to let it sit for about 10-15 minutes, allowing the DNA to precipitate out. Any questions? All right, so I'll give you about 10-15 seconds to scrape the inside of your cheeks. No, just with your teeth. Just scrape the inside of your cheeks. You're trying to shed off some of the cells. Okay? Unmask. Put the water in your mouth. Ready? All right. Go now, and I'll time you. Now spit it back out in the cup. Okay, put your mask back on. All right, so I'm going to come around before we go on to the next step and bring everybody a piece of paper towel, because, again, you're going to have that on your thumb. Give you something to wipe it off with. After we're done, you can go wash your hands also. Okay, so the last class, the breakage, there were very few that were actually successful in precipitating out a decent amount of DNA. That could be for many reasons. I'm just assuming that they didn't swish hard enough in the mouth and they didn't bite the inside of the cheeks. All right, so you're going to go ahead. You're going to take this. Now, kind of see what I'm doing here? How I pinch the side here to kind of make it a corner. You want to do that so you eliminate spilling any on the table. So pour it in. Okay, then put the cup off to the side. All right. So next step, we're going to take our lysis, uncap it. Remember, we're only pouring 5 milliliters in. So pour slowly so you don't over pour. Five on the dot. So, yeah, up to the 5. Yeah. Oh, it was windy. Okay. Oh, you weren't supposed to pour it in there. Oh, no, never mind. No, you're right. I was looking at your other hand. Okay. So you could put that back in the air. So, you have yours in? Yeah. Only 5 mLs though. So only half the amount. You good? All right. So go ahead and put that back in the beaker then. No, not that. You keep that. Put the other one back in the beaker. Okay. Now cap your thumb over the top because you don't want to spill any out. And we're going to do this, not shaking it. Do it very slowly because you don't want to create a bunch of bubbles. We're going to do this for about a period of 2 minutes. Yeah, you're not mixing. Look at me, like this, all the way upside down, because it's not mixing. Yeah, like this, completely upside down. That's why you're capping it, so you can do that. See how mine is one color throughout? Yours is still 2 separate colors. Your soap is at the bottom. Yeah. Oh, that's okay. It's a little bit. You gotta know. You gotta make sure your thumb's over the top completely. It's nice. Yeah, I'll show you what mine looks like. Yeah. See how when you look at the side, you see my thumb. That's not part of the thumb. That way, that creates a seal, so I'm not losing anything. All right. So you do yours. You guys take your test tubes now. You're going to place them back in the beaker because we got to try to let some of those bubbles settle down. In addition, we got to give the lysis a couple minutes to work. So clean off your thumbs. Are you having any luck there? Now, I would do it for you, but I'm not because that's your saliva. So, um, let me see if I have a stopper that might help you. You know my other one. Still not having any luck? Alright, here. Why don't you back with your palm there? Make sure it's tight. No. Top the beaker would be like this. Yeah, make sure you. Oh, you know what? No, you should still be able to do that. Yeah, why don't you take this and stir it around? Slowly, okay? And then go ahead and lift it up. Yeah. But get down all the way to the bottom too. Alright, so do that for a couple minutes. Next is going to be the alcohol. So in about another minute, I'll bring the alcohol around to you. Remember, it has to be cold, so that's why it's still setting on ice. You should be good now. All right, so just put everything back in your beaker. You'll wait 2 minutes. Let the lysis work. You could put that also. Good. All right. So I'll come around, bring the alcohol. And you can come up in a minute, rally, and grab one, okay? You just need a little bit more time. All right, so again, this is the step where you can screw everything up if you end up mixing them. So again, at an angle, pour in very slowly. Oh, wow. That's a decent amount. So let that sit, allow more DNA to precipitate out. There we go. Now just follow along when I do the next step. So again, we're going to fill with alcohol. You're going to take your scoop with this end, the hook. Now remember, when you stick it inside the test tube, you don't want to mix the layers together. So do it slowly. Don't go past the middle layer, okay? So mine actually, I don't even have to go to the middle layer on mine because my DNA is at the top right here. 1 second. And now what you have is a DNA necklace.